Okay. So it should be 10 o'clock now. Can everybody hear me? You can write in the chat or... Okay, good. So I will keep an eye on the chat. And also we have the... Okay, great. And also we have the Q&A, which you should be able to find in the bottom, I think. And so let me just start sharing. So one thing I was told that the meeting is recorded and it will be published on the I2K YouTube channel. So I think if someone has something to say about that, should reach out to the organizers. So yeah, Krista was actually writing in this chat about that. Okay. So let me just start sharing here my screen one. Okay, let me see if I can also open the chat. Good. Now I have chat and Q&A open, so if you write me, I should be able to see it. So I was asked to share the presentation. I don't have a presentation. I will simply go through the software. For the relevant links, I thought about it, basically would be the publications that I'm talking about. So that I will definitely send them by the end of the meeting. I don't have them right now, so I will maybe do it later. But I don't have a presentation for the meeting itself. The way I thought I would do this, I will go through the software, the Cell-ACDC software, and I will basically live demo what you can do with the software. It's kind of dangerous because things can go wrong, but I hope everything will be fine. We already did it in the past and it was fine, so we hope it should be fine. So maybe a few sentences to introduce the software, introduce myself. My name is Francesco Padovani. I am a postdoctoral scientist at the Helmholtz Zentrum in Munich in the group of Kurt Schmoller. And we are a lab which is a mixed lab of computational scientists and cell biologists. We mainly work with yeast cells as model organisms and chlamydomonas. But with the software, we kind of expanded into collaborations with other people using other kind of organisms and other kind of imaging modalities. What is Cell-ACDC? Cell-ACDC, you can see it here. I hope you can see the sharing. You should see the documentation basically. And this is the link for the documentation. And I will actually send all these links later. And basically, Cell-ACDC, it's a GUI-based software. So it provides a graphical user interface. And for the segmentation, tracking, cell cycle annotations, and quantification of microscopy data. What does that mean? It means that you can perform the task of segmentation. So you can detect your object of interest, like single cells or whatever other object you have. You can track these cells over time, and you can annotate the cell cycle. And by cell cycle, I mean like mother-daughter relationships. And so like cell lineage pedigree, and you can quantify the whatever other fluorescence channels you have. So you can extract measurements based on the intensity of your channel, or also morphological measurements and all kinds of other measurements. So the way Cell-ACDC works is by having multiple modules, one module for each task, and a main graphical user interface. So the main thing about ACDC is that it allows you to run models that the community have developed, like CellPost, StarDist, whatever. And then it provides you a user interface that allows you to correct the segmentation outputs, correct the eventual tracking errors, and annotate your microscopy data. So it's designed for basically allowing you to visualize all the steps of the analysis, to go through, for example, if you have time-lapse, to go through all the frames and make sure that everything looks correct and everything is it should be corrected in case it needs to be corrected, and then you can also annotate on top of this. So the main advantage of using ACDC is the time-lapse support. We have a strong focus on supporting time-lapse microscopy. You can also use it for three-dimensional like Z-stacks or two-dimensional data, that's for sure, and we have actually users doing that. But I think that the main advantage comes when you have to work with time-lapse data. And time-lapse data that you want to be sure it's 100% correct or as correct as it can be. Okay, so here, this is the documentation to install it. It's a simple Python package. We have here the instructions. And if you plan to use ACDC in the long term, I recommend spending a little bit more time to install from source. So you can definitely install the stable version, but chances are that you have this can correct, and we get the to give you updates and give you new features and whatnot. So you can see here 3 ways to install. And from source means that you pull directly, you clone directly the code from GitHub, and then you install it as any other software. We will not cover the installation. It should be quite well explained here, but if you have problems with the installation, don't hesitate to reach out to me. And actually something I will share later is a link to our Slack workspace, because the way you can reach out to us is of course from the Image@C forum, for those of you who are familiar with this. But for those of you who don't want to have their requests publicly available, essentially because it's a forum, they can contact us directly on a Slack workspace that we have already with many users. Good. So one thing, we are open for collaboration. So if you think ACDC does not work well yet for your data, but you see some kind of potential, feel free to reach out and we can discuss how to implement new features and work with you on the data that you have to analyze. Okay, so this is very quickly an introduction. So we should actually see the software in action now. And the way you run ACDC, actually you already have it running here, but let me close it. Once you have it installed, let me just get a new one here. Once you have it installed, it's a Python package. So you run Python packages typically from the terminal. So here I have my terminal. If you are on Windows, I recommend using either PowerShell or the Anaconda prompt. We cover these steps on the installation instructions, so I wouldn't worry too much about it now. And the way it works, you activate, if you have Conda, you would activate the environment where you have the software installed. And as I said, I will not cover these details because we have them in installation. So for those of you who are not too familiar with Python packages, I recommend first getting a little bit familiar with that. But then it's simply an installation. Then you just run one command in the terminal. So you run the command ACDC. And when you run the command ACDC, it will basically open the software. You don't see the animation because it's on the other screen, but you would see a logo. And then you see the software appearing. And in the back, there is also the terminal, which also will communicate. But we try to communicate through the GUI to the user and not much through the terminal, so that you don't have to keep looking at both. You should get pop-up messages and stuff like this. But sometimes it happens that the errors are not displayed properly, so they might be in the terminal. So I still recommend to keep an eye on the terminal every now and then. Okay, this starts. You get this launcher here, which is it shows all the modules plus some menus here, which we'll see later. And the way it works is that you start typically with some microscopy data. So like you have your raw microscopy file, which could be from Carl Zeiss, could be from Nikon, could be from Delta Vision, whatever microscope you are working with. And hopefully you should be able to create the data structure starting from this module. It's a little bit tricky because there's so many formats for microscopy data. So even though this step should be the easiest one, sometimes it's kind of the first barrier you find. So you want to be able to convert the microscopy data into the structure that is required for Cell-ACDC. And I will talk about the structure later, but hopefully if everything goes well, you should be able to use bioformats here, which is this fantastic library that allows you to open like more than 150 formats from microscopy files basically and automatically convert them into the required data structure. If that fails, we also have Fiji macros, so that because bioformat is written in Java and running it in Python is not that easy sometimes, so we also provide Fiji macros that allows you to do this automatically from Fiji. And if not, as I said, reach out and we find a way to work with this. I will not show this today because it's simply you would go through a wizard of selecting your file and saying how many positions you have, how many times, for example, how many frames you have and stuff like this. And hopefully, if everything goes well, you should get this kind of structure. So here I prepared some data, and actually this data, it's all published in papers, so I will also share later some links to this data directly. So basically what we have, let's look at some data here, not annotated. Okay, so the way it works is that you will create the files in what we call positions. You can see here position 6. If you have multiple positions, you would have position 1, 2, 3, 4, 5, whatever positions you have. And positions are typically, I don't know, a place that you imaged over time. So for example, if you start from a single cell growing over time, that will be your single position. I think in Fiji they are often called series, or they could be the tiles, but basically are portions that you acquired with the microscope and field of use basically. And inside the position folder, so you can have as many positions as you want, and typically organizing your data into experiments. Let's say on the day of a specific, you run 10 positions, you will have that folder. In this case, I called it not annotated, not very informative, but you would call it your experiment 1, for example, and inside you might have multiple positions. I prepared only one position here just for today, and inside you will have an images folder, which is where all your data created by CDC will end up. And the reason why we have a subfolder is that we are working on other software that will have other outputs. For example, software for other type of analysis, they would end up outside of the images folder. So images is for ACDC and other software will be outside here. So inside the images folder, you will get a TIFF file where you will need to have one TIFF file per channel. So if you have four fluorescence microscopy channel, you will have 4 TIFF, all separated, and the TIFF file it can be either a 2D file if you have 2D data, it can be a 3-dimensional Z-stack inside the TIFF because TIFF supports multiple images, or it can be a 3-dimensional in terms of 2D over time, or it can be 4-dimensional in terms of 3-dimensional, so Z-stack over time. So these are all the dimensions with support. Plus, you can have as many channels as you want. So you also have multiple channels. In this case, there is only one channel. But if you look at some other data that I prepared for today, for example, the time-lapse 4-dimensional, we see that we have, oh, why is it not details? We see here that we have MK8, M-neon, and TPMT. So these would be 3 channels. And this metadata is data that is created by the module that you see here, which creates this metadata. The XML is simply the data as it is, the metadata as it is saved by your microscope. The CSV file is more structured, ACDC specific data, which I can actually show you here. We see description and values, and we have the numerical aperture, dimension order, size D, which is the number of frames, number of Z slices, the name of your channel, some wavelength, and some stuff that has been extracted automatically from the raw microscopy file, so that your data always goes together with some metadata that describes whatever you have acquired. And okay, so you get these TIFF files that, as I said, you can create with this module, or you can create with Fiji macros, or you can also create manually if you have other ways to create this. So you will need one TIFF file per channel. Okay. Modules that you see here, once you have your data, are the data prep module. And in the data prep module, you basically go into this if you have to select, for example, let's say you have 4-dimensional data or 3-dimensional data that you want to segment on a projection, or you want to segment on a specific z-slice, or you want to segment a specific z-slice over time, because that's also what we do often. We segment only a z-slice out of our z-stack. So then you would have to tell the software which z-slice or which projection you want to use. So you would launch this data prep module, and you would load some data. So here, let's load, let's say, I have it here, I2K data. Let's say we load yeast time-lapse, not annotated. We select the position folder. And actually, this is not, it will be, let's do 4-dimensional data actually. You select the position that you want to load. In this case, it's just one position. You select the channel that you want to visualize. In this case, I would load the TPMT, which allows me to see the cells, while in these other two channels, I have other structures. And it takes some time because this data is a little bit big. You can see it tells you have 65 frames and 24 z-slices in a 512 by 512 XY image. So here is just pop-ups that appears. You go through this wizard essentially of confirming, and here we see our data. Here we have a scroll bar for the frames, and here we have a scroll bar for the z-slice. And in this case, you basically go through the data. Let's say you like z-slice 13, and you simply look at all the frames and see that z-slice 13 looks kind of fine, because here it will be out of focus. Here it will also be out of focus. So slice 13 is now the one we want to use, and everything is saved automatically. So you don't have to do anything. You just visualize and select which z-slice is the best for you. Because if I, for example, say here I want a 9 or 8, when we go back to 13 and we go back here, it remembers you used z-slice 18 for frame number 26. So then when you go into segmentation, it will segment that exact z-slice. And the other thing you can do in the data prep module, you can crop your images. So you would, and you can align over time. So for example, I think this is already aligned data, probably not, I forgot. But basically you can align, let's say your data moves around over time. We have an alignment routine. You would click on this button here, start process of aligning the data over time, so that it might be helpful for tracking, for example, because if the cells move because of the microscope movement or drift, it's much easier to correct than trying to track them when they are not aligned. Whether it works for you or not, that depends on the data as usual, but it's worth a try. So in the data prep, you can select the z-slice, you can align the data over time if you have time lapse, you can select projections. Here you see kind of projections. You have max projection, mean projection, or median projection. We say a max projection, for example, if we prefer the max projection, and then you would segment max projections. And the other thing, you can crop the data. So if I would press start now, it would go through the process of aligning, but for the interest of time, we don't do this now. But basically you would then get a crop ROI so that you can crop the data and make it smaller. So this is kind of the data prep you would do. You can skip this if you don't need any of this. If you don't need to align, you don't need to select these slides, you simply skip this step. Okay, the second module is the segmentation module. However, you often don't know which model works best for your data. We have many models. We have Cell-Pose, we have Stardist, we have Segment Anything. So you don't know exactly which model works for you. And you would go into this module. This is for batch processing. So let's say you have multiple experiments, multiple positions. You want to segment them all with Cell-Pose because you know it works well. So you would run this segmentation module. And I can actually show you for the not annotated data, because I already know that the model that I want, but I will show you later what would you do if you don't know the model yet, because it would be a kind of a waste of time to test one model on all of your images. So you would like to test them on specific frames, for example. But anyway, let's see the wizard we got. So here I have some not annotated data, which you select. Again, you get this image properties dialog, which you always get just to be sure it's always correct. You press OK, and here we see the models. So here we have the models that we have, and we have them, each one of them, most of them has a publication related. So on our documentation, I think somewhere like here in citation, here in citation we have the segmentation models and the trackers. The reason why is in citation because it's really important that you don't cite only ACDC. If you use Cell-ACDC in Cell-ACDC, you should cite both Cell-ACDC and Cell-ACDC. So then here you have the models, you click on them and you get the publication related to that specific model. Now, this is very to make sure that you don't give credit to where credit is due. And here you see the models. Some of them might be custom for my specific Cell-ACDC implementation. We have Omnipose, we have Easy, we have Stardist. And in this case, I know that Easy V2 for me is the best model because it was specifically developed for yeast, and we're working here with yeast data. I will have some other type of data later. And we select the model, and we should have a look at the terminal in this case because it says importing Easy. And one thing happens is that if you don't, so ACDC doesn't come with all these models pre-installed. So if you run Easy and it was never installed, you will first go through the installation process in the terminal. It will automatically install that specific model. Sometimes it doesn't work directly. Sometimes you have to restart ACDC, but usually it should be fine. Most of these models, I think all of these models have been tested all together in environment, so it should be fine. There is some problems, for example, with Cell-ACDC, you cannot have both V2 and V3. So if you install V3, ACDC will first uninstall V2 and install V3 and the other way around, but you shouldn't care too much about this stuff. Hopefully it should be automatically done by ACDC. Once you have your model here, you get the parameters of the model. So you get this dialog. You see Easy V2 parameters. The pre-processing is standard on any model. So you can apply as many pre-processing steps as you want. So sometimes a Gaussian filter is helpful, or we have hot pixels, ridge filter, spot detection filter. So it might help the model to better segment your structure, and you can have as many steps as you want. You could have first a Gaussian filter plus a ridge filter or whatever. You would simply add multiple steps here so that the images before being segmented go through this pre-processing. I know that I don't need pre-processing because my data is actually already working well. Here, there's the model type, has Easy, has a phase contrast model type, a bright field, a fission yeast. And there's some parameters for the segmentation, and there's also some post-processing. The post-processing is for filtering out in the after segmentation. I don't know, let's say you want to keep only the objects that have a specific size. So then here you would say minimum area 10 pixels, minimum solidity, max elongation, plus all kinds of custom features, which we'll talk about later about the measurements. So I will not show you now, but basically all the measurements that ACDC computes can be used to filter your object data. So let's say you want only the brightest objects being segmented, then you would, I don't know, select only the objects whose mean intensity threshold. So then you automatically get those objects filtered. I think I don't need post-processing actually with this data, so I probably remove that, but I don't even think I will run the analysis anyway. Maybe the first 10 frames to show you. Okay. Here we give a name. You can have as many segmentation files as you want because you might segment, I don't know, one channel with one file and one channel differently. So you can use Cell-ACDC for fluorescence and Stardist for whatever else. So you give it a name. In this case, I can call it, I know it's the phase contrast channel. So I can give it a phase contrast name here. and this is the base name. So this is something important. Any file generated by ACDC will start with this base name here. And actually I can show you, I think we are looking at this data here. I forgot which data I opened. Yeah, exactly. So here you see any file that will be created will start with the same base name underscore the rest. So this is the dia ph3 is the name of the channel, phase contrast. And here we will get a new file called like this, segmentation phase contrast MPZ. And the segmentation files are MPZ formats. So these are NumPy Python specific formats, but we have conversion tools, which I will talk about later. We have utilities to convert these into other file formats, let's say TIFF files to open them in Fiji and to do other kind of post-processing. So we try to be integrate. So we try to make ACDC can be integrated into other software. Okay, here we select okay. It will ask you whether you want to segment a subregion with a ROI, with a region of interest, like a square ROI. In this case, I don't need to, but if you want to, you can. We say no. We say up until which frame we want to segment. And I think for the sake of the time here, we do only the first time frames. Let's see how fast they are, but otherwise we can just stop it and go into, because I already have it prepared. So we say we segment the first 10 frames, and here we said track object. Okay, here we can choose a tracker. Basically, you can choose a segmentation model and a different tracker. We have these trackers implemented, which you can actually see here. They have their own publication. We have Delta, which is for like bacteria cell, deep sea, tapir, Bayesian tracker, TrackPy, and these are available here. However, there is a thing to say here. If you plan to manually correct tracking errors, the tracker right now is kind of not meaningful in a sense that we track the data anyway in the module we will see later. So you might also not need to track the data now because we have a real-time tracker in the other module, which we will see later. So it might be actually not needed to track the cells. However, if you have a good tracker that gives you good enough data, you can just select that tracker and get your data already tracked automatically. So in this case, I choose easy, even though I know I might have to fix it later. And here we get this dialog whether you want to save and run the workflow later, which means it will save a file that you can take and run this workflow without the graphical user interface onto a cluster, for example. So let's say you have access to GPUs or high performance environment, computing environment, cloud system, then you would be able to run ACDC without the graphical user interface, because often these environments cannot have a graphical user interface. All they have is a terminal. So then you would save and run it later, or you can just simply run it now locally. In the tracker, we will see some in the terminal plus here, we will see some process, and basically this should go through the 10 frames, which should take fast enough so we can actually wait and see the results. And yeah, you will see, as usual, I always recommend keeping an eye on the terminal just to have or just to be sure that everything is going well. But it should be, everything should be exactly. Okay, so here we have final dialog for 33 seconds, segmented these 10 frames. And now the segmentation model is closed. And if you had a good model, you had a good tracker, you might be already ready to go into some downstream analysis where you use your data. And as we will see here, now the phase contrast MPZ file has been created. So this is now the segmentation file. So now we can finally go into the main module, which is where we spent most of our effort, allowing you to have a framework so that you can correct the output of these models. And this is the third module, which we simply call the GUI. And here, as usual, we go to the open folder button here. You could load directly an image, but as we said, I'd rather recommend structuring your data properly so that the ACDC can take care of everything, and you simply load a folder. So here you would load a folder. We select this folder that we just segmented now, and here we see it asks us to select which segmentation file to load. So this would be the phase contrast segment. If we have multiple, we select which one we want to load. Again, we get this dialog of the image properties, because here you start seeing physical size, micrometer per pixel, which might not be relevant for you now, but it might be relevant later when you want to know the cell area in micrometer square. You might have had the wrong one all the time, but then when you actually care, you want to edit that. So you simply get this dialog all the times. You can always edit the metadata. And now we see here the GUI. The GUI is made with these two images. They are the same exact images, the same exact image, but they can be annotated differently. So you see here with the scrolling wheel on my mouse, I can zoom out and in. And here we see that the same image, but annotated differently. So let's say we can have a clean image here with do not annotate. We see here the two ways to differentially annotate the two images. We can have the segmentation masks, we can have the contours, we can have the IDs. In this case would be the same. So typically I like to have the segmentation masks here on the right and the contour on the left, but it's up to you, whatever you want to have. And this is helpful to keep an eye on two things at the same time. Of course, you can play with only one image if you want. You would right click on the right image, I think also on the left image, and you see you can simply untoggle here, show duplicated left image. So you would work with a single image here. I like that, so I still do it. So we do show duplicated image. Here we see some adjustments for the intensities. For those of you familiar with Fiji, it's like the adjust brightness and contrast dialog, where you adjust the minimum intensity and maximum intensity. And they can be differentially adjusted right and left, so you can have different kind of visualizations. You can choose the color map with right click. We have some color maps. Let's say you want flame or whatever. You can have all kinds of color maps that might make sense for fluorescence microscopy data, might be easier to see. You can make a custom color map and then save it, because here you can edit this color, for example, and have a different, save it and have your custom color map. But I go back to grayscale here. And this is the visualization part. Here you have the frames scroll bar. This is two-dimensional data over time, so we get only one frame. And we see we segmented the first 10 frames. And actually we see some mistakes from the model, so I think it was actually easy v1. One can get better results. But basically, we see that we segmented up until frame 10 and not frame 11. Now, here is where you can see that you can test different models. So this is just the viewer mode. Here we see we are in viewer mode. If you try to do anything in viewer mode, like segment, it would tell you, look, you are in viewer mode, you should change mode first. So we need to change mode, and we have these modes here. So we have the segmentation and tracking, which is where you correct and track cells over time. So these have been already tracked. So you see that cell ID 2 stays cell ID 2 over time. Actually, let's start from frame 0, frame 1. So these have already been tracked. And if there is any new ID, for example, any new object, it says here there is a new object. And this is now a mistake from the segmentation model. I will show you later how we can fix this, but you get some kind of visual help and information. And it will tell you that for ID 4, for example, has been lost. It was here in previous frame, and now it's lost here. So you have this visual help from the title and from the ACD, from the frame, from the software. So now we can go into segmentation and tracking. And the other modes that we have are statistical analysis, where you basically annotate This is more specific for yeast. Some people have been using it for other model organisms, but basically you can annotate. For those of you familiar with yeast data that you can see here, this is a mother cell and this is a bud. So we want to annotate this relationship mother with buds, and we want to annotate when the buds divide from the cell, for example. And this applies also for cell division of what we like to call symmetrically dividing cells. It's not really symmetrically dividing, but it's just that visually the mammalian cells divide like standard cell division. It's one mother cell dividing in the center into two daughter cells, and the mother cell does not exist anymore. It's a cell dividing into two. With yeast is different. The mother cell still exists. It simply makes new buds, basically. And this cell cycle analysis is that what allows you to pair these, to annotate mother to daughter and the daughter like the sisters, for example. However, since this is very specific, more specific for yeast, it can be adapted to mammalian cells, to symmetrically dividing cells. We now have a new mode called normal division, which is normal, like standard cell division of the mother cell divided into two cells. But we have to go into these modes only when we are 100% sure we have correct segmentation and correct tracking of the cells over time. You get all kinds of weird errors because let's say this is now a cell 4. ACDC would like to unpair this cell. It's a new cell, so it's probably a daughter cell. It should be paired to the mother cell, but of course this is just a segmentation error. So it should be fixed before trying to annotate the cell cycle. Okay. So here we go into segmentation and tracking. We should start from frame 1. And in segmentation and tracking, you get these tools. These tools are like a normal brush where you can draw new cells, erase, where you can erase the cell. And everything you do in ACDC, most of the things you do in ACDC should be undoable. So we have Control Z to go back to what we had before. And that's kind of a big help because we always make mistakes in correcting stuff. So it's good to have an undo function. We have a pen tool where you can draw with like this. And we have a magic wand, which works more like for intensity, like nuclei, where you click and it should segment the entire nuclei. And we have all kinds of tools. These tools are all documented with the tooltip. So you leave your mouse on it and it tells you what you can do. So for example, this one is copy lost contour. Basically, when there is a lost object, the contour will be visible from the lost object, and you can tell I want to keep it, so then it's not lost anymore. It appears back into your frame because it was actually not lost. It was simply the segmentation model didn't segment it at that time. So all the tools that are available here are meant to perform tedious tasks in like fastest way possible, while still keep an eye on the data, while still being able to look at your results and correct them. As I said, if you have a good model, like a good segmentation model, a good tracking model, you might be fine with not correcting. But if you want to start annotating cell division, for example, it might be beneficial to actually look at the data and visualize it over time. One thing that confuses some of the users at the first time they use ACDC is that here, scroll bar appears to be at the end. The reason why it's at the end is that we never checked in this segmentation and tracking mode the future frames. So we never went into the second frame with this mode, okay? And this is what it says, is the last checked frame is number 1, which is also the maximum of this progress bar. If I click here, of course, I can go to the next frame. When we go from one frame to the other with segmentation and tracking. So actually there is a question that I like to address first. Can you track 3D data and can you do the correction of 3-dimensional track data? You can, but not all the functions are available in 3D. So the way we are adding support for 3-dimensional data over time, I work myself with 3-dimensional data over time. but not all the functions are available because they need to be discussed with the users. The reason is that, for example, let's take simple brush. What should we do with the brush? Should we brush on only the visualized z-slice, or do we want a brush that is able to draw on multiple z-slice? The brush is already active, it draws only on one z-slice, but this was just an example of those functions. Of some of the functions, they need to be discussed. So for example, even just a simple delete object. Do we want the delete object function to delete only the z-slice, or do we want the delete object to delete the entire object? Because we don't see whether on the other z-slices, so there are things to be implemented for three-dimensional track data, but you can definitely load three-dimensional data and track it over time and test it. Some of the trackers already work in 3D, like TrackPy and Bayesian tracker, I think. But if you need a specific function, reach out to us and we discuss how we can implement this further. It's the most annoying data, three-dimensional data over time, but it's definitely doable with some patience, of course. Unless, as I said, you have a tracker that already works well and you're fine with all kinds of errors it makes, or it might not make errors if you're lucky. And the reason is actually that you can have your own segmentation model, you can have your own tracker. So if you have a segmentation model that works well, you can plug it into ACDC. We have instructions how to do that. I think they're also explained here. Segment displayed frame, for example, add a custom model, and you get instructions. I need instruction, or I have the model. Let me select it. You can basically add your own segmentation model, add your own tracker, and benefit from the framework of ACDC to correct that, or run it essentially if you prefer to run it directly. So here, I hope, which is the fast way to select a whole object? How do you save your changes? Okay, so changes are saved once you click save. So they are at the moment only on RAM, okay? And to select the whole object, you don't have selections of object in the sense that you don't need to select object unless you have to do something with it. So for example, you want to delete an object, we have a function for that. So it's customizable. On my system here, I click the scroll wheel button. So here I click on cell 3, which I'm not, no, I think I actually customized it. Customize keyboard shortcuts. It's actually shift left click, because here you can customize all the shortcuts. So I need to press shift left click to delete the object. And you can customize this and delete the object. If the brush, for example, it's kind of smart, so it recognizes you are on top of an object. So it recognizes you want to edit that object. So you see the brush here, it would make a new object. You do shift left click. But here it recognizes you are onto an object. So it's already selected for you. So you don't have to select objects. You can, of course, select objects in the brush by removing this auto ID function where you select the specific ID. But here it recognizes that I'm already on a specific object and we can edit this object, make it bigger, for example. So we can do Control Z and go back. The selection, actually something useful here. The pairing of colors here is kind of unfortunate. These are two different objects, but the same color. But we can choose a different color map for the masks. So then also the color of the brush when we hover onto an object is kind of more recognizable than before. And if not, if you still like this map, but they still don't make sense, you can press Shift S to, you see it's randomly shuffling. Okay, this actually looks better. Randomly shuffling the colors to have better colors in terms of pairing. So then now brown and green are better. And when we see the brush goes over and it changes the color. For the eraser is the same. If I erase here, I'm erasing everything, both objects. But if I start from a specific object, you see the other object was not touched. So I was just erasing only one object instead of going on to the other object. Okay. Now, one of the main thing of ACDC, one of the main advantages of ACDC is that it tries to simplify the corrections. So let's say you have to change, there is a tracking error at some point. Cell ID 1 becomes cell ID 3. You want to be able to change that into cell ID 1. ACDC will now on keep tracking that cell with cell ID 1 and not 3 anymore. So all the future cells that have been tracked wrong as cell ID 3 will be automatically corrected to 1. So we try to reduce the number of actions from the user by having these frameworks, these algorithms in the back running for you. And this is what you see here called real-time tracking. So you can choose the tracker you want to have in real time. And that means that every time you go to a new frame that has never been visited before, you will get automatic tracking of the cells of that frame. And this is why I said at the beginning that choosing a tracker might not make any difference, because you might have to adjust and then all of a sudden the tracking is wrong. So you have to retrack the data. So you don't have to retrack the data manually yourself. You simply have this real-time tracking happening. So then when you go to the next frame, cells are tracked. If instead you want to just view the data, make sure you are in the viewer, because otherwise things happen here and you don't realize. And this is the reason why this is the last check frame. So we should go to the next frame only when we are 100% happy with this frame. So we corrected everything, everything looks fine, so we go to the next frame. In the next frame, we realize that there is a new object, ID 6, which is not good. So I do shift click, delete. Okay, so then we can go to the next frame. This frame looks good. And if there are no new objects, no lost objects, here it will tell you looking good. It might not be true because it might be wrongly segmented, but at least according to ACDC, there are no lost objects, there are no new objects. Now here it says there is a new ID. Let's say now I didn't realize this new ID. I go next, and this is now tracked, but now it's lost. So now it says, oh, where is my ID lost number 6? Okay, I am at frame 7, and I did not realize there was a segmentation error, but I realize it now because I get this bright condor here. It says lost ID 6, and it asks where is 6. So you can go back here, let's say, do shift click, because it's my delete object, and now you get asked. So now it realizes you are correcting a frame that was supposed to be already fine, which you can do, of course, because we all make mistakes, we all miss tiny corrections we should have done. But now we are allowed to apply to future frames. So we want to delete this number 6 everywhere because we just made this mistake. So we can choose to apply only to this frame and reinitialize future frames. So let's say we delete only now, and we want to make the future frames as they were never seen. But that's not what we want here. We want to apply only to this frame, and keep future frames as they are. So we want to delete it only now and still keep the information that future frames were visited. But what we actually want to do, we want to apply to all future visited frames. You can also apply to all future frames, including the unvisited ones. It means including the ones above 7. But now in this case, it's kind of dangerous because we don't know whether cell ID 6 in the future is an actual cell that we want to have. So we shouldn't delete cell ID 6 for all future frames. We should delete them up until we visited, because it's when we realized, okay, this is not a right object. So we say apply to future visited frames, and we see that now it's gone on all future visited frames. And something that you get from ACDC is that when I'm scrolling visited frames, like here up until 7, nothing happens from real-time tracking. So everything is kind of disabled. It's more like a viewer here. And the reason is that you don't want ACDC to screw up your corrections. So you don't want the tracker to retrack cells that were correct. So up until frame number 7, in this case, nothing will happen. However, when I'm frame 7, I go to frame 8, things happen again. So we have cell number 6 again. This is a constant mistake from the model. It thinks here there is a cell. So in this case, there are some other tools that might help. You might want to just go to the future frames where you see, oh, 6 appears, I should delete it, and then apply all. But the other way is to have a constant deletion ROI, which we place here. And now any object that will appear here will automatically be deleted. So it can be a square deletion ROI. Like this is a deletion ROI, but it can also be a custom deletion ROI, which actually doesn't have a tooltip. Okay, so I have to add a tooltip here, but basically it's the same as the deletion ROI. But the idea is that you draw your ROI that, because square might not work, you might draw a little area, because this is kind of annoying. When models make mistakes, they're very consistent with that mistakes, and they keep doing it forever. So this is very helpful. Here, any object appearing here will be automatically deleted, but it will not be deleted permanently until you save. So if I move this away, the object reappears. Because now at some point there might be a true cell here, and I actually want it. So I can remove this, the cell appears back. If I put it on, the cell is gone. Once I save, it's actually deleted forever. If I don't want deletion ROI at some point, now it's always here. Exactly, there was a cell actually here. If I don't want this ROI at some point, okay, let's say here I don't want it anymore. At frame 9, I do remove ROI, the object appears again, and now it's gone, but it's still there in the previous frame. So from now on, I don't want this deletion ROI anymore, because now my cells actually grew into that place. So we don't want to delete cells there. We say we don't want that deletion ROI anymore. And from the future on, from that point on, you will not have those deletion ROIs anymore. In this case, it was actually correct to have them, but this is just an example. Okay, so this is very quickly what you could do in segmentation and tracking. You basically go through all the frames and correct them. If you don't have time lapse, it's a little bit easier, of course. You can use the brush. You can use all kinds of tools here to adjust your segmentation. You don't need to deal with tracking, so you will not have real-time tracking. You will not have all this kind of stuff. But I think we spend a lot of time with time-lapse data, so we spend a lot of time to making ACDC useful with time-lapse data. For other type of data, I actually have examples later. I should keep an eye on the clock. Okay, 10:50, we still have time. Okay, now for the cell cycle analysis, I might go through the cell cycle analysis later because not everyone here is yeast people. So let's just skip that for now. We can come back to this data later. Now I want to show you other type of data. Unless there are questions, but okay. So as I said, it's not only for yeast data, it's for any kinds of data that needs to be corrected, for example. And you can have, for example, we have, and I will send you the links about publications. Let me just write it here later. Maybe we finish 5 minutes earlier than the 11:30, so then I have time to write actually, I think I can also reach you, all of you, by email. So I will send you also the publications link to this data. Because, for example, we have Mario Vitacolonna at Mannheim University here in Germany, who is working with organoids. So because ACDC can scale well also for a lot of cells or for a lot of frames, for example. And he has this kind of data. I just want to show you before we look at the rest. He segmented the nuclei here. And actually, this is a good example of, so here, maybe just to explain this dialogue, it says there are 3,000 objects, 3,700. Depending on your computer, that might be a lot. So you might want to switch to low resolution annotations. In my case, my computer is powerful enough and I can stay on high resolution. And basically the text in high resolution looks better, of course. In low resolution, it's still readable, but it's much faster. So it might be more relevant for your computer. And this is a good example of what people can do with ACDC in three-dimensional data. Here we see an organoids. Maybe I should not annotate the cycle because it doesn't make any sense. And here maybe we don't annotate the IDs. You see here, this is a z-slice. There are 146 z-slices of this organoid. I think this is more like a spheroid. And what basically Mario has been doing, he has these organoid images in a collaboration with Merck. They're testing different kind of drugs. And what he wants to do, he wants to segment the cells and extract measurements in 3D. This is kind of another advantage of ACDC, is that if you have three-dimensional data, you can have three-dimensional segmentation masks, and then you will be able to compute three-dimensional measurements. So here I can show you the data. We see it's quite complex spheroids data. The reason why it's very well segmented is because they trained CellPos on their own data, so they don't actually have to do corrections, but they still use ACDC to extract, to visualize, of course, whether it makes sense or not. There might be actually mistakes, but in the big picture, they don't care. So it actually looks pretty good, to be honest. And now when you zoom out, it looks like a mess of numbers because you need to zoom in and see the numbers smaller, of course. The white color is a little bit unfortunate here, so we can actually change the color of the text and maybe make it, I don't know, black or blue. And white was actually pretty okay. But yeah, you can change the colors with right click. There's all kinds of tweaks to the visualization you can have, which I will not go through, but feel free to test them. And here, it's a good example of having multiple segmentation files, because you have seen me at the beginning when I loaded the data. Mario has now multiple channels. He has the nuclei stain, Ki7, Ki67, YAP, and the membrane stained. And then he has multiple segmentation, one for the Ki67 and one for the nuclei. And what you can do in ACDC is essentially when you have multiple segmentation files, they might be of the same structure. Let's say you have segmentation of the nuclei and segmentation of the cells, but now they are independent. So you don't have the same ID, for example, of the nucleus to the cytoplasm. So what you can do in ACDC, and this is one of the reasons why Mario was using it, we have utilities here to do all kinds of mini batch processing automatic workflows. And we can convert file formats, for example. This is why I said at the beginning I would go back to this. You can convert your MPZ files to all kinds of other, like to image ROIs, for example. You can convert them to import them into Fiji. You can convert them to TIFF files, and we have conversion files here. And these utilities work on multiple experiments, multiple positions in batch processing all at once. So if you have to convert a lot of segmentation files into TIFF, you would just run this on all of your files. But something interesting here, we have tracking and lineage. And in tracking, we have this utility called track or count subcellular objects, which means you assign the same ID as the cells they belong to. So let's say you have nuclei and you have cells. You want to be sure that the cell with that nuclei have the same cell ID, so that when you extract measurements, you will get a row for the nuclei ID 1 and a row for the cell ID 1, which are the same, they belong to the same object. So they are the cell with its own nuclei. And this is what Mario has been doing here. He's been segmenting the nuclei, the Ki67, and then track them so that they are the same object, so that you can extract measurements from all channels based on nuclei mask, and from all channels or whatever channel you want based on cytoplasm mask. And when you do this, actually, here it asks you to select the experiment folders. So you would go through this kind of wizard. I want to do it on the organoids, so I do it on position 1. We can select additional experiment folders if we want to do more folders in one shot. We say no here. And this is what you can do. In tracking mode, you can delete the subcellular objects that do not belong to any cell. So if nuclei don't have any cell, you can delete them. You can delete the cells that do not have any subcellular object. So cells that don't have nuclei, for example. You can delete both, or you can only track the objects, and whatever is without a subcellular object or the subcellular object without the path will stay there. But the useful thing is that you can create a third segmentation, which you can call, for example, I don't know, cyto would make sense. And cyto now would be the difference. Oh, I clicked again. The difference between the nuclear segmentation, the cell segmentation, and the nuclear segmentation. So you will get only the cytoplasm with the same ID of the nuclei and the cell, but then it will be essentially the cell with a hole so that you can extract measurements only from the cytoplasm. So you can have intensity of the nuclei, intensity of the cytoplasm, and intensity of the whole cell. So I will not run it now because we actually already have it here. But basically, once we have this, we get these two masks that are describing the same object. Now, once you have this, you go to the measurements menu here, and you have set measurements. These are all the measurements that ACDC can compute, plus whatever custom measurements you have. So you can have your custom measurements in terms of, you would have to write a little Python function that takes as input the image, the segmentation, all kinds of inputs. It processes it and it spits out a number, which would be the measurement of one cell. And that would appear here. You can have all kinds of custom measurements. So here, for example, in the custom measurements, I have the CV, which is the coefficient of variation, which basically means I want to take the mean and divide the standard deviation of the signal divided by the mean of the signal to get the CV. I have a little Python function that I can automatically add to ACDC so that you compute those measurements. We loaded the nuclear mask here. Yes, we loaded the nuclear mask, which means now we get measurements based on the nuclear mask of whatever channel you want. Here we have the Ki-67 channel, YAP metrics, and membrane metrics. If nuclei metrics don't make sense, we don't save them. If YAP metrics don't make sense, we don't save them. And each one of these measurements come with an info button. So you can click, let's say, mean of max proj, kind of self-explanatory, but let's say you don't understand what it is. It would tell you that the mean is computed from the pixels inside each segmented object. So this is the mean of the signal. However, since you loaded three-dimensional Z-stacks, Cell-ACDC needs to convert the Z-stacks to 2D images using a max projection in this case. So when you see mean max proj, this is the mean of the cell, but from max projection image, which could be helpful, could be not. So for example, for confocal images, a mean projection makes sense. From epifluorescence images, the sum projection makes sense because you sum all the light. No, sum projection. In that case, only the Z-slice would make sense. So, and the metrics like mean, sum, amount is pretty useful because it's basically the total fluorescence intensity automatically corrected by the background. So there are also metrics that are background corrected. I will not go through all the info buttons here. I suggest you do it and try to understand. But of course, if there's something that doesn't make sense or is not clear, just text me. And basically, it computes all these measurements, mean, sum, amount, concentration, mean, median, max, quantiles, from max projections, from mean projections, from the specific Z-slice. And this is the Z-slice that we selected in data prep. So in that case, the Z-slice most in focus for epifluorescence can make sense because the epifluorescence kind of captures a lot of light from neighboring Z-slices. And if you have three-dimensional data, also directly from the three-dimensional object. So this is the mean of the 3D, which in this case, it doesn't make sense that it tells you that it needs to convert because this is actually three-dimensional measurement. So this is the mean from the three-dimensional object. And it's pretty helpful. For example, if you have cytoplasm, nuclei, and cell, you can get the mean intensity in all of these compartments for all of the channels. So these are the measurements, and you can have custom measurements for each one of the channels. Like here, the CV, it's applied to all channels. If you have to save it, you save it. Plus, we have some physical measurements. We have the cell area in pixel, the cell volume in voxels, and these are, there's a little bit of a difference between these two. This one is computed from two-dimensional mask, which is rotated in 3D, which in some cases we showed in our publications that it's fine. It's still good compared to actual three-dimensional. But if you want to be more accurate, you might want to segment your cells in 3D and get the actual cell volume in 3D. Both in voxels and in femtoliters, and this is where those pixel sizes that you get asked all the times comes into play. If you want to have the cell volume in femtoliters, you want to have the correct physical size, the pixel size in micrometer per pixels of your image. Here we have morphological properties, all kinds of morphological properties that, like, for example, major axis, minor axis length, whether you want to morphologically describe your object. One thing you can do, you can add combined measurement, where you basically get these little calculating where you can combine all of these measurements. Let's say you need the mean of the channel nuclei minus the mean of the channel Ki67. Then you go here, say nuclei, cell signal, mean max proj, minus Ki67, cell signal, mean. Okay, we give it a name. This is my mean nuclei Ki67. You need to test its output to make sure it makes sense. It tells you that 3.9 minus 1.1 is 2.8. Looks correct to me. We say okay. And now we get this measurement here. So here we have mixed channels, combined measurements, because we are mixing channels. And if you forgot what it was, you press info. It tells you that it's the nuclei mean minus the Ki67 mean. And this will always appear now here all the times for this specific data set. But if you don't want it anymore, you simply delete it and you don't have it anymore. So these are the combined measurements. And this is basically what you can do with the measurement stuff. But now we did everything on a single organoid. Let's say we have thousands of organoids. We want to be able to compute these measurements in batch processing on all 1,000 measurements. This is actually what Mario has been doing. We want to go, in this case, for this kind of batch processing, you should always think in terms of utilities. The utilities are for batch processing things that don't require any input from the user. So we go to the utilities menu, we go to the measurements menu, and we say compute measurements for one or more experiments. And here is the same. We select as usual as before, the experiment folders. We have this position for this organoid. If you want to select more, you select more. If not, we say no. We select which segmentation mask we want to use for this kind of measurements. We say the nuclei, and we get the same dialog. We set this dialog once, and it will run for all the experiments, all the positions that we selected. So you get all the measurements straight out of this. And the other, so for the utilities, this is something that some users kind of miss on, because we're working on the documentation, but it's hard to get exactly all aspects. So this is why I think this workshop is useful, because then we already covered a lot of things that if you would go through the documentation by yourself, you probably would miss. So we have the measurements in terms of segmentation. There's some kind of stuff. I don't have examples directly here, but what we choose that we want to stack two-dimensional segmentation objects into 3D objects. So we want to apply the same Z-slice segmentation to all the Z-stack for some reason, because you get like cylindrical shapes that might be enough for some analysis. Because 3-dimensional segmentation is painful. If for those of you who work with it, we know it's hard to correct. It's hard to make sure it's so. If we can go If we can cheat, let's say, and use two-dimensional masks, why not? Okay, so these are the utilities, and this is the modules. This is a new software that which we have. It's called Spotmax, but we don't cover it today. Maybe in the next workshop, or maybe I will record a tutorial on that. So these are more or less all the modules. So if there are no specific questions, we still have like a 10 minutes. And what we can do, we can go back. Let's see what I prepared in terms of data. Okay, I can show you some other application from some other users. So we have people with organoids, people with yeast, like all our lab and other labs. Can you run this headless in the cloud? So the segmentation module here, yes, you can, because so ACDC can be installed headlessly, so it can be installed without the GUI. You can use it as a Python package if you know Python. Otherwise, you can set up the segmentation module workflow locally and run segmentation and tracking on the headless on the cloud. And because at the end, I don't know if you saw it at the beginning, but basically at the end of the wizard of setting up which position you want to segment, which model you want to use and stuff like this, you can save this to a workflow file and then run the workflow file from the terminal with ACDC -p and path to this workflow file. And then it will run in the terminal headlessly. This is the only part, except for being a Python package. We don't use it much as a Python package, to be honest, but sometimes we do if we want to import Cell-Pose from ACDC because it has slightly different implementation. But this is actually the only part that you can run because all the other modules, if you think about it, require input from the user. So you cannot run headless to run. So you could potentially run the measurements headlessly. That's true. And if this is something you need, we can implement it pretty easily. But most of the things cannot be run headless because they require input like brushing, correcting segmentation errors or stuff like this. The measurements, actually, I thought about it now. You could set up which measurements you want to save, which positions you want to save, which experiment folders you want to run it headlessly. So if this is a feature that that you might need, feel free to write me or open a GitHub issue, and we can make most of these utilities basically running headless. But the framework for running headless is already there, so it will be very easy to add. Okay. So what are the biggest data set this has been applied to? Have you ever applied to 100 terabytes of 3D data, for example? So no 100 terabytes of three-dimensional data. And I would, so as long as you can load the single position into the RAM, then it should work. In sense that if you can split your 100 terabytes into positions that can be loaded into RAM, then it should work. Because loading positions is sequentially, so they will be loaded, discarded, loaded, discarded, loaded, discarded. So far, we worked on data. So the data is the channels are saved as TIFF files. TIFF files cannot be lazy loaded, so they need to be loaded fully onto RAM. So this is one of the limitations, and this is something we want to work on in the future. We want to add support for OMISARs and lazy loading so that you can use this with large data sets. We didn't have, so for example, if you already have 64 gigabytes of RAM and you split your data into positions, typically people have these large data sets, but when you look at the actual data that you need to load, let's say one video of one channel, it's really hard to have a really big data set of one channel, one video, for example. But yes, the main limitation is that the files need to be TIFF files, so they need to be loaded into RAM. It seems better to use bioformal in Cell-ACDC how to export. So maybe I read the questions correctly. It seems better to use bioformal file in Cell-ACDC how to export bioformal file from Leica LASX. So we have people using Leica. Leica is kind of annoying because they keep making new files. I'm not sure why they do it, but I think we had one user recently with some super recent Leica files and it worked. It seems better from ACDC in the sense that if it works, yes, it is better. But often it doesn't work because it's hard to run. Not all systems, like on Windows, it's fine, but on macOS already, Java Bridge doesn't work well because we are using Python bioformats. So in that case, I recommend macros that we have. I can actually show you quickly. If you go to our repo on GitHub, we have here Fiji macros. And we have a couple of macros to automatically create the data structure starting from the Leica file. If you can open the Leica file with bioformats from Fiji, the macro will work. If not, then you need to. There are other so you might have to. Some users had to do it directly on the microscope software because then Leica has support for their files, hopefully. So then you can convert into TIFF files there and then use bioformats, or save into, I don't know, OMI TIFF, for example, if your software supports it. So there are some options. It's annoying because it's one of the easiest, it should be one of the easiest steps, but for some reason in the bioimage analysis community, that's what happened. Probably because it's easy to build microscopes, so there are so many manufacturers that make their own file formats, but I know there are massive community efforts for OMIZAR, for example, to get new microscopy manufacturers to get on with using OMIZAR. So hopefully the future is a single file format for all the microscopes, but we are not there yet. But none of our users had to give up on that step. They all could somehow convert their files into TIFF files. Should be doable. Actually, Mario replied to you. Mario is the scientist behind the organoid data that I just showed. Okay. So now, one last thing, maybe just 5 minutes, is with the time-lapse. Maybe I show you quickly data from Ciro Salino. He is at the TUM working with T cells. And he's using ACDC a lot actually to segment, track, and annotate cell division of the T cells, because of course it plays an important role in T cells differentiation, for example. And here we see that ACDC scales well also with a lot of frames. Here we have 1500 frames, not many cells because it starts with one single cell. Eventually they divide and become many cells, but not immediately. So, but yeah, the main limitation so far, which is something we want to work on, is that the TIFF files needs to be small enough to be loaded all into RAM. If you have RAM issues, smaller portions, or you contact us and we can actually discuss how to implement OMI ZAR support, for example, for lazy loading. And here we see data from Ciro. He's working with these T cells. We see these T cells. This is a single T cell over time, and they divide quite rarely, so there's not much happening, to be honest, but they divide and he wants to segment them and track them. And he found ACDC is the only thing that worked, because as you can see, these cells move like crazy. Trackers tend to have a hard time with these cells moving a lot. And this is actually stuck here now, so it's kind of easy, but if you see there, cell number 1 is kind of wandering around everywhere and moving absolutely crazy. One reason is that the time frame is every half an hour here, so it's a lot. It's a big step, so they actually move not that much if one would image them every 3 minutes, but still they move a lot. And we found ACDC to be working well in terms of essentially manually annotate this data, because none of the trackers that are available works well. Good. One last thing that we have time for, since we didn't have any bug or errors, we should push our luck. So one thing that I did not cover was the time-lapse annotation, set cycle annotation. So that should be doable in these last 5 minutes. We actually segmented the first time frames here before. So here we have this data. As you remember before, we did not save, so we should save this time. So we should go quickly through these deletions here, which were the only thing to be done. And I'm doing it now quickly manually because I know the problem, but I could have done it with. One thing is that if you miss and you click on the background, it asks you. You clicked on the background, enter the ID that you want to delete. It was actually the ID 6. It gives you already the closest one, because in this case I missed the cell when I clicked. And here we are at frame 10. Good. So here it shows me that this is a never segmented frame. So we should probably reinitialize here. Because one thing you can do, you can always reinitialize the last annotated frame, the last visualized frame, the last validated frame with this button here. So now we can go to cell segmentation, to set, which it would be more like annotations of mother and buds. It reminds you, it actually finds out that the last annotated frame is frame 1, so we actually never annotated this frame. So it asks you whether you want to go back to that frame or stay on the current frame. We say yes, we go to frame 1. And in this case, for yeast cells specifically, we want to pair these two objects. And let me see if this bar can be moved away. Okay. We have this button here. Every button has a shortcut, so at some point you might get fast at activating functions. Like I don't need to go there and click. I know it's A, so I can click A and the button is now active. And basically it tells me I need to click on this button and release on this one. And now these two are paired. And this is a yeast specific thing that I will not go through, but basically we want to segment the buds separately, but still pair them. And here it tells you what you did. So for the first frame, you confirm. And we are annotating basically this cell cycle stage. These are now cells in SG2M, because this is a yeast specific thing. When they are budding, they are in SG2M. When they are not budding, they are in G1. It tells you the relative ID. So cell ID 1 is related to cell ID 2, and cell ID 2 is related to cell ID 1. The relationship cell ID 1 is a bud, and cell ID 2 is a mother cell. And if this was normal cell division, it would say sister and daughter and mother. And it tells you in which frame the bud emerged. It cannot tell us because it was already emerged before we started imaging. And then the vision frame, which I want to show you now. We just say okay, and we go through the data. Now, there's not much happening in this data set, but basically what you can do is to annotate division. So let's say now this bud at some point divides. I don't think it does in these first 10 frames, but let's fake that it divides here. So we right click on the cell, and now it's annotated as divided. And from now on, it's divided from the mother cell. If we have to correct this, we can go back. Oh, let's say actually it divides here and not there. We go here and click here. And now it's automatically fixed. All future frames have been automatically corrected to allow you to click only once. So there's a lot of automatic propagation of corrections. So you correct once and hopefully don't have to correct that same thing over and over multiple frames. Okay. So this was more or less everything I could cover in 1 hour and a half. And if you have, I will now find a way to send you links. Basic question, what is a loading position? So a position, so here we load. Okay, let me just get the file as they are. Okay, here what we loaded is this dataset, yeast time-lapse 3D, and not annotated. And a position is, in this case, for me, a single cell. I usually acquire multiple positions each position is one single cell. So in the end, I might get 10 positions, 30 positions, 40 positions, whatever. And each one of these positions is one field of view, basically. The field of view is your position, and you can have, I think in Fiji, they're called series, and basically, or tiles, if you make multiple tiles, but it's your single field of view. How do you do, thanks for the memo, sure, no problem. How do you do the measurement over time? So the measurement over time, if you have time-lapse data and save the measurements, ACDC will go through all the frames you annotated and extract the measurements and get the table in the end. In the end, you will get a table. So here you go set. Actually, I did not show how to actually do the measurements. When you do save, let's say we want to save what we did, you get asked, do you also want to save the measurement? Here you still have a chance to do set measurements. Or if you say yes, it will simply save the measurements. And it asks you up until which frame? We say up until frame 10. It goes through the saving. So it goes through each time point, and for each object computes the frame, the measurements. At the end, it asks if you want to concatenate multiple tables, because one can end up with 10 experiments, 40 positions per experiment, but you want a single table, so you can concatenate them automatically in ACDC from all your experiments, all of your positions to one single table, where there will be a new column called experiment folder name, experiment folder path, and position, so that you know which measurement is coming from. Here we say no, because we have only single position. And now, in the folder, we have a new file called ACDC output phase contrast CSV. And the CSV file, I can open it here, kind of messy, but basically you will get your annotations, like the cycle stage here and stuff like this, but you also get the measurements, auto background value, median, and stuff like this. So this is a table you can load into Excel, Python, MATLAB, whatever, because it's a CSV table where each row is one cell from one frame from one position. So the single object. And as you could see here, you also get the frame column. So this is frame number 4, cell ID number 1, and you get the measurements. Okay, when I create the third segmentation file for only the cytoplasm, I will get the same number ID of the specific, yes. So when you do that, you will get the same cell ID of the nuclei and the cell. That's kind of the thing of tracking. You track the nuclei to the cell, they get the same ID. And then the cytoplasm, if you do create the third segmentation, because not always we need this third segmentation, the third segmentation, which will be the difference, they will have the same cell ID. So then you can get measurements from the cytoplasm, from the nuclei, from the cell, of all the from all the channels. You just need to make sure that you load the correct segmentation file. And something important for that is that we, if you remember, we call the segmentation file segmentation phase contrast MPZ. So the measurements specific for this file will be in the file with the same name, ACDC output, instead of segment, phase contrast. So if you then have segmentation nuclei, segmentation cell, segmentation cyto, you will get 3 tables, 1 cells, 1 cyto, 1 nuclei, and inside the tables, they will all have the same ID. So if, for example, you open them and join them, you can have a single table with all the measurements. Sure, no problem. Okay, so I think we are now done. And I will send you the links. I think there is a way to reach all of you that joined the, I will ask that anyway. And I will send you the link to the documentation. You can find it anywhere in Google, Cell-ACDC docs. I will send you a link to join our Slack workspace if you plan to use ACDC and you want to write directly to me. I will send you maybe and the links to the publications of the data I showed, so that we give credit to people that created that data. Good. Thank you very much, and enjoy the rest of the day and the weekend, which is soon. Yeah, it's Wednesday. Okay, great. Thank you very much. Bye.